Light pollution. The effects of light wavelengths on melatonin production in the Drosophila melanogaster and the correlation on memory. Light pollution, also known as photopollution, luminous pollution, or sky glow, is excessive, misdirected, or obtrusive artificial light in the night sky. This causes the night sky to become brighter. This map of the Earth was taken by a satellite. It shows the amount of light that is directed into the sky. More dense populations have a greater amount of light pollution. These two photos were taken by satellites. The photo on the left is New York City and the photo on the right is Hong Kong. These two cities give off the most light pollution out of all the cities in the world. In the United States, 6% of the electricity produced is used for outdoor lighting. Of that, 30% of the light is wasted as light pollution. This wastes about $10.6 billion a year. Streetlights are one of the worst structures for light pollution. In the diagram, it shows different street lamps. The lamp all the way to the left shines light in all directions. This causes the most light pollution. Then from left to right, the light becomes more refined to a certain spot and causes less light pollution. The top shows what the night sky may appear like with the different lights. The organism that will be used in the experiment is the Drosophila melanogaster. A Drosophila melanogaster is shown. The reason it is used is because of its small size, short lifespan, and its close similarities to the human genome. Drosophilas also produce melatonin, which will be measured in the experiment. The wake-sleep rhythm of humans is synchronized by the intensity and spectral composition of light. Light pollution can disrupt our circadian rhythms and cause many human health problems, such as sleep deprivation and even cancer. The reason for our alertness and our drowsiness is because of the production of melatonin. Melatonin is secreted by the pineal gland. This gland, as the picture shows, is located in the brain near the top of the medulla oblongata. Melatonin is secreted to make us fall asleep. This is regulated by our circadian rhythm, which was shown by the previous slide. This photo is a picture of the brain of the Drosophila melanogaster. In the brain of the Drosophila melanogaster, melatonin is synthesized by proteins that were created. Such proteins are AANAT1. This picture compares the circadian rhythms of the Drosophila melanogaster and humans. They have a similar circadian rhythm. Many basic biological, physiological, and neurological properties are conserved between mammals and the Drosophila melanogaster. This graph from the National Coalition Against Breast Cancer shows the number of breast cancer patients over the last 50 years. The x-axis is the year, and the y-axis is the number of cases. The line shows that over the years there has been a steady increase in the number of breast cancer cases. This slide shows the visible light spectrum and their wavelengths. The red light has the largest wavelength, making it the least powerful and intense. Then from left to right, the wavelengths become shorter, making the light more intense. The violet is all the way to the right, making it the most intense with the shortest wavelength. This is a photo that was put up by Cinzano, 2001. It shows light pollution in America over the past 60 years. The map shows that in the late 1950s, little light pollution was present. But they then predicted in 2025, areas like New York City will be over 81 times the normal light level at night. This will cause the Big Dipper not to be present. This chart is from an experiment conducted by Stevens, 2009. He wanted to see the probability of breast cancer in women who work the night shift compared to women who work normal hours. He concluded that women who work late hours are more susceptible to breast cancer because their circadian rhythms are thrown off and melatonin levels are changed. He also conducted an experiment that linked light pollution to melatonin fluctuation, which he also linked to a higher probability of breast cancer. In the article by Alb, 2013, they attempted to find the wavelengths that most affected certain bodily functions. One of the bodily functions that they tested was melatonin production and which wavelength most affected it. They found that melatonin production was most affected by wavelengths from 430 to 680 nanometers. The purpose of the experiment is to find which wavelength of light will have the least impact on the production of melatonin and on memory. Based on the studies of Stevens 2009 and Selzer 1981, it is predicted that the blue light will have the most impact on memory and melatonin production. This is the flowchart and the methodology for the experiment. These are the variables and the controls that are going to be used in the experiment. The control is a regular clear LED light. Then there are 5 variables. Each variable is a different light, which is a different color. The colors that are going to be used are red, blue, green, orange, and yellow LED lights. All of the lights will have constant amount of wattage and each will be identical to each other, other than the color. There are 3 major procedures that will be used in the experiment. The first procedure is how to take care of the flies. What is first needed will be the vials, which can be ordered on Carolina.com. Then in the vials, there will be food placed on the bottom. The food will be made from the Drosophila medium mix made by Carolina. All that is needed is to add water. Then place 10 milliliters of the food into the vial and place flies in the vial and put the cap on the vial. When genders need to be separated, put Drosophila on a flat white surface and sort with a brush. The melatonin levels of the flies will be found by first crushing half of the male population of one variable and the same with the females. Then the crushed flies will be centrifuged at 20,000 Gs at 4 degrees Celsius for 20 minutes. This procedure will be done for each variable. When tubes are taken out of the centrifuge, melatonin levels will be measured by a melatonin ELSIA kit ordered from Antibodies Online. The rest of the flies that are not centrifuged are taken to a t-test maze to test memory. The t-test maze on the screen is the maze that will be made for the experiment. Flies will be tested by first running a preliminary test to make sure that flies travel to the lighted tube. The lighted chamber is indicated. Then, to test the fly's memory, filter paper will be soaked in quinine solution and placed in the lighted chamber. The flies that will pass the test should reject the lighted chamber at least 5 out of 7 times out of 10. Data will be analyzed with a one-way ANOVA post-hoc Scheffé test with a significance of less than 0.05. The Drosophila melanogaster, Drosophila vials, Revolutionary Science Centrifuge, Disposable Centrifuge Tubes, Parafilm, Instant Medium Blue, Aluminum Foil, Quinine Solution, Dome Light Fixture, and Incandescent White Light were all from Carolina. All of the light bulbs needed were bought from Bulb America.